In this protocol, we detail an in vitro model of human tauopathy. This method fills a materials need which may be useful for drug screening and disease mechanism studies. The main advantage of this technique is that the cell culture paradigm allows for a more rapid assessment of tau toxicity and potential tau therapeutics compared to animal studies.
To begin this procedure, thaw the basement membrane matrix coating for culture plates at 4 degrees Celsius, make 1 milliliter aliquots, and store them at -20 or -70 degrees Celsius. Next, reconstitute the basic fibroblast growth factor in sterile PBS at a concentration of 10 micrograms per milliliter. Make 10 microliter aliquots and store them at 4 degrees Celsius. Then, set out a new unopened 500 milliliter bottle of diamium F12 with glutamine. Add 10 milliliters of B27, 5 milliliters of N2, and 5 milliliters of penicillin streptomycin to prepare neural stem cell media. Place 50 milliliters of the NSC media into a conical tube, and add 10 microliters of the prepared BFGF. Store this NSC plus BFGF media at 4 degrees Celsius. First, remove an aliquot of frozen basement membrane matrix coating for cell culture plates from the freezer and allow it to thaw at 4 degrees Celsius. Add 385 microliters of the thawed basement membrane matrix.